For the final set of talks, what we designed for this year's Norman Bethune program is to have a series of talks that are dedicated to structural biology. Structural biology is a very important aspect of the science that we do at the CBR, and perhaps to some degree may have been a little bit under-celebrated, but no longer. That is no longer going to be the case. So our first speaker in that regard is Clint Spiegel, who's visiting us from Western Washington University. Dr. Spiegel completed his undergrad and master's degree at Oregon State University, followed by his PhD at the University of Washington and the Fred Hutchinson Cancer Center in Biochemistry in Seattle. Followed that by postdoctoral training at the University of California, Santa Cruz, and he'll be speaking to us today on the structural analysis of the intrinsic tenase complex. So Clint, thank you very much for coming.
All right, thanks everyone for sticking around. First, I want to thank the organizers for the invitation. It's an honor to share the stage with so many accomplished scholars and physicians. And the work I'm going to present to you today is an ongoing collaboration between our lab at Western Washington University just across the border and the Doering and Lawler labs at Emory University. And what I'm going to tell you about is what our lab studies, or what we use, are multidisciplinary structural approaches to study complexes in the blood coagulation cascade, namely factors 8 and 9. Okay, so many of us are familiar with the blood coagulation cascade, and I won't belabor too many of the details here, other than to remind everyone that we have two intersecting pathways, the extrinsic and the intrinsic, that converge on the catalytic activation of factor 10 to 10A that ultimately leads to thrombin and then an insoluble fibrin mesh. What our lab has been focused on for several years is looking at one of the central regulatory reactions in this pathway that is called the tenase complex, or the intrinsic tenase complex. And so if I talk a little bit more about the intrinsic tenase complex and what it's made up of factors 8A and 9A. And so shown here is a diagram of factor 8A at the center, sitting on a hypothetical membrane surface of an activated platelet. And this is something we've studied biophysically and structurally over several years that I'm not going to talk about today. But what needs to happen is that this protein has to serve as a scaffold from which this other protein, factor 9A, comes to the platelet surface and then catalytically activates 10 to 10A. Factor 9A by itself is a relatively poor catalyst, and the presence of factor 8A increases its catalytic efficiency by about 200,000-fold. And just to remind everyone, genetic deficiencies in either factor 8 or factor 9 lead to hemophilias A and B, which affects 1 in 5 to 20,000 male births worldwide. So in order to understand our structural talks, we need to talk just a little bit about the domain structure of factor 8. Okay, factor 8 is synthesized as one long polypeptide chain, largely in the liver, made up of 3 A domains, a B domain, and 2 C domains. As it's processed and secreted into circulation, most of the B domain is cut out. And in fact, we can remove it entirely for protein therapeutics. And once factor 8 is activated to 8A, it turns into a heterotrimeric complex. Now, there are some significant complications for this processing of factor 8. But what we can say to start with is that the canonical treatment for for hemophilia A is therapeutic infusions that we call factor 8 replacement therapy. Some problems that occur with this is that it's very hard to make, and so it has poor biosynthetic efficiency. It gets trapped in the unfolded protein response in the ER. It has a very short half-life in circulation and has a potent immunogenicity that is more immunogenic than most proteins that we see. More to the point, once it's activated, it becomes rapidly inactivated through A2 domain dissociation as well as activated protein C cleavage. Okay, the other side of the intrinsic tenase complex is factor 9, which is one of several vitamin K-dependent serine proteases. And so I don't need to belabor the details here, but as it becomes activated in circulation to form factor 9A, we have a light chain and a heavy chain, the heavy chain having the protease or catalytic domain, and then we have a GLA domain and 2 EGF domains. Okay, and so again, what happens is factor VIII becomes activated. It dissociates from this big multimeric protein von Willebrand factor. And in the presence of both 8A and 9A, they bind to the activated platelet surface to perform this 10-ACE reaction. And what else I could say about 9A is that it also possesses a relatively short half-life in circulation and has somewhat of mixed immunogenicity that is complicated by the common, well, not common, but the repeated observation that these antibody inhibitors can result in anaphylaxis. So before I get to sort of 3 vignettes of the studies that we've done in the last few years, I wanted to just comment briefly on the current state of hemophilia treatment, which is both promising as well as complicated. So to start with, I had mentioned this half-life issue for both of these therapeutics, and it turns out that through protein engineering approaches, we've been able to make therapeutics that have significantly longer half-lives, and this is largely due to things like Fc fusions or pegylation to the proteins. But maybe what's more transformative for hemophilia treatment are non-factor or bypass therapies. And the most notable ones that are used today are bispecific antibodies that mimic factor VIII's function by binding to factor X and factor IXA, bringing them together for the 10-ACE reaction. And so those are called emicizumab and MIM8 are the clinically approved ones. But maybe more to the point, what's been observed is that what's really important in treatment for diseases such as hemophilia is that you just really have to balance hemostasis. And this can actually be done indirectly, which has been shown to be effective recently by using anti-tissue factor pathway inhibitor monoclonal antibodies. Where treatment for hemophilia gets complicated is in our current state for gene therapy. We have both great successes as well as some mixed observations. And so the great success comes from a liver-directed adeno-associated virus delivery of a factor IX gene that is hyperactive in the presence of factor VIII. And that is a variant that was found naturally in the population in Padua, Italy, about 18 years ago. And so this mutation increases that activity by 7 to 9-fold, which is required to overcome liver transaminitis due to the AAV delivery. There's a similar approach that is now clinically approved for factor VIII gene therapy to treat hemophilia A, but it is less effective. It's quite expensive, and the community is not quite sure if this is exactly the right treatment for them for a variety of reasons, and so it is being discontinued towards the middle of this year. And then lastly, we have ongoing issues still of anti-factor VIII inhibitors, which is a significant complication leading to pathogenesis for factor VIII replacement. And so if I start there for a moment, I should point out that we see factor VIII inhibitors develop in approximately 30% of hemophilia A patients that receive repeated infusions. Most of the epitopes that are pathogenic occur in the A2 and C2 domains, but we commonly see them in the C1 domain as well. This can be overcome by bypass reagents, as I just mentioned, or immune tolerance induction. But what else can be useful for this is porcine factor VIII, which is also an effective therapeutic and is now clinically approved as a recombinant therapy. And so the recombinant porcine factor VIII was developed by Chris Doring in Pete Lawler's lab about 20 years ago. And what Chris had noticed in doing so is that porcine factor VIII seems to behave much better than human factor VIII. It expresses better. It has a longer half-life once it's activated, presumably because it has a tighter affinity for the A2 domain. And so because of that, Chris and colleagues decided to try to humanize the porcine factor VIII by coming up with cassettes to find a human porcine hybrid that mimics those superior qualities. That's a protein called ET3. And what ET3 is a chimera B-domain deleted therapeutic where the porcine sequence is in the A1 and A3 domains and the remaining structure is human sequence. So it's 89% human sequence. And subsequent to this development, Doring and colleagues showed in a mouse model that they could use a lentiviral-driven vector to express ET3I transgene in hematopoietic stem cells and show sustained levels of factor VIII. And so this is, we came into play sort of parallel with this development to do some structural work with the ET3 platform. And so with this, we determined the structure and published it in 2020 of the highest resolution structure of factor VIII to date. This is notable because all of the pre-existing structures sat between 3.8 and 4 angstrom resolution with some significant errors in its model due to its low resolution. Okay, and I'll touch more on this in a moment. But what we can do with a structure that provides more of an atomic level of detail is we can start to see specific mutations that exist in the hemophilia population that mechanistically inhibit factor VIII's function. Okay, so for example, shown here lining how A2 binds to A1 and A3 is that you can, these particular point mutations result in increased A2 domain dissociation. What's more is we can also be structurally informed about bioengineering efforts that had previously been published. So for example, mutating interfacial residues to cysteines allow for us to anchor the A2 domain onto the remaining factor VIII structure to provide more stability and a longer half-life in circulation. We can also increase the hydrophobicity of that interface, and so shown here are some residues that are mutated to valines or isoleucines in order to do so. But what else was unique about our structure is that it was crystallized in a space group that was different than all of the other factor VIII structures previously determined. And so because of that, we have two separate views of the factor VIII structure. The A model, which we could call the canonical conformation of factor VIII, overlays very well with previously determined structures of human factor VIII. And the B model shows rotation about the C2 domain in a left-handed barrel-like twist. And this is not the only observation we've seen like this. I'm not going to report on all of our antibody structures today, but several of our structures of factor VIII in combination with inhibitor antibodies show conformational heterogeneity for the C2 domain specifically. Okay, so most recently, Doring and colleagues completed a clinical trial where they used the ET3I expression cassette in an ex vivo system using a lentiviral-based system to transduce autologous hematopoietic stem cells. And in doing so, show sustained factor VIII activity over several months to up to 2 years. While these numbers are dispersed, in every case there is measurable factor VIII activity following the ex vivo treatment. And maybe what's more dramatic about these patients, so this is 5 individual patients, is that when you look at their annualized bleeding rates and annualized joint bleeding rates, both before and after the lentiviral ex vivo treatment, you see that it drops precipitously. So showing that we have durable expression of factor VIII with this type of platform, which may lead to maybe a more robust gene therapy approach for treating hemophilia A. Okay. As I had mentioned, structural characterization of inhibitor epitopes is something we have been working on for several years. And this is important not only for understanding the immune pathogenesis of these antibodies, but they also allow for us to understand mechanistically how factor VIII works. And so what you can see here is that anti-factor VIII inhibitors will in vitro inhibit factor VIII's procoagulant function by blocking its ability to bind several of its binding partners. And so to date, we've used a combination of X-ray crystallography and cryogenic electron microscopy to determine several structures of factor VIII bound to antibodies that are notably anti-C domains, where we've seen both the C1 and C2 domains. And this allows for us to have a better understanding of the structural nature of that immunogenicity, which largely has surface-exposed basic residues as part of the epitopes. It also allows for us to refine a model for how factor VIII interacts with both the von Willebrand factor as well as the lipid membranes of activated platelet surfaces. And so the data that we don't have to date are structures of factor VIII bound to the other highly pathogenic antibody response, which is notably to the A2 domain. And what I could also note is that while we've determined some structures of antibodies that have been isolated from hemophilia patients with inhibitor, a lot of our structures come out of a murine model. So the one antibody structure I'll talk about today we'll call NB11B2, which is anonymized from the patient from which it is isolated. And so this is from a severe hemophilia A patient that has a very potent inhibitor. And there's not much published on this, which is why we are referring to a patent from several years ago. And the only structural information from the patent has to do with peptide inhibition studies that show that a peptide at 484 to 509 would be the epitope predicted for this particular antibody. And we already know that this is an immunodominant epitope based on prior data from several years of research. Okay. Our colleague from the NIBSC in Oxford, UK, had decided to take several of these patient-derived antibodies and recombinantly produce them, which is our source of these antibodies. And what Carmen showed in her clotting assays is that these antibodies, or this one in particular, NB11B2, is very potent and inhibits the 10-ACE complex to completion quite rapidly. So shown here is our 2D classes of the NB11B2, which we call a Group A anti-A2 domain antibody inhibitor, of which we have 5 different groups that have been previously categorized by Pete Lawler's lab. And we also show that its inhibition reaches saturation rather rapidly. What's notable is that antibodies that overlap with this epitope have very high Bethesda titers, between 10 and 40,000 Bethesda units per milligram. And what I can also say is that we have another antibody that we're studying that has an overlapping epitope that also has these properties that we refer to as 4A4, which I'll return to in a little bit. So when we determine the structure of NB11B2, as shown here, we determined it to 3.7 angstrom resolution. And when we look at the epitope, we see that the epitope of NB11B2 is as expected, and it centers on the 501 to 505 loop of the factor VIII A2 domain. But immediately when we looked at this structure, and we know full well that it's highly potent, it was a bit befuddling to see that where this epitope is not directly where we would expect factor IXA to bind factor VIII, leading to a variety of hypotheses, right? For example, does this block binding to factor X? Does it have some other allosteric effect? And so on. And so when we look back on the literature, what we find is that what buttresses between the antibody epitope and where we predict factor IXA to bind is a basic patch. And so when this basic patch is mutated, so for example, if we mutate arginine 489, 490, and lysine 493, then it does have a significant impact on the ability of factor IXA to perform its function. And in doing so, it has no effect, not much effect on the KM, so it still seems to bind factor X fine, but has a significantly reduced turnover number for that complex. But what I've done recently, and this is preliminary data, is to use an emergent technique called mass photometry. And if you have not seen what mass photometry is, it's a technique that measures particle size distributions in dilute solutions. And it really, for me, I found this technique to be somewhat unique because it is relatively hard to structurally study and mechanistically study these proteins at the concentrations that they really are in circulation. Right, so factor VIII's circulation concentration sits at about half nanomolar, half to 1 nanomolar. And so what we first saw here, as shown on the left, is you see 2 distributions. One is with ET3, so a structure of factor VIII that should be more stable than human factor VIII. And between 5 and 10 nanomolar concentration, you find that it starts to fall apart. Okay? And so intact factor VIII goes from being 70% to 50%, and we see a combination of the light chain and heavy chain, which are about the same size, that increases. So we were surprised by this, but we were perhaps even more surprised when we used this antibody 4A4 that overlaps with the NB11B2 epitope, in that what happens when we add the IgG is that it seems to cause dissociation of the factor VIII heterodimer, so even before it becomes activated. So shown here in the lower right, we see a monoclonal antibody, and the only way for this monoclonal antibody to be in this size distribution is if it's stripping off the heavy chain, which it recognizes. Okay, so in the last part of my talk, I briefly want to talk about our preliminary data on the intrinsic tenase complex, which again is a combination of factor 8A and 9A. And what we find is that these are hard to study because of the rapid dissociation of the A2 domain following activation. They're both highly flexible proteins and hard to crystallize in their own right. And if we were going to look at this in some sort of catalytic, meaningful way, we should be doing it with lipid membranes. And I should note that that's not used in this study. Instead, what we use is our quiver of antibodies. And so we collected a cryo-EM data set with an anti-C2 domain antibody as a fiducial marker to allow for us to readily orient the tenase complex. And so when we do so, we're able to reliably split these particles apart into both an activated factor VIII set of particles as well as an intrinsic tenase set of particles that allows for us to see not only the catalytic domain, but the EGF2 domain. And so if we first look at the structure of activated factor VIII, you can see directly that the A2 domain is starting to dissociate on the grid. And so if we tally up by just saying we have inter-domain connections of 5 angstroms or less, that we see a total of about 16 contacts that are severed upon activation. But perhaps what we're most interested in is looking at some of the structural features of the tenase complex. And so shown here in purple is the catalytic domain with an all-important helix called the 170 helix, which is where the Padua variant exists. So Padua variant is this R338 or R170 position, if you're using chymotrypsin numbering, that is mutated to a leucine that causes this increase in activity. And so we see this electrostatic interaction with the arginine residue, and our expectation, or our hypothesis, if you will, is that if this is mutated to a hydrophobic residue like leucine, that it would actually allow for the helix to be pulled further into the factor VIIIa A2 domain. Something that perhaps may substantiate this model is in a recent ASH meeting, there was a report of a novel factor VIII mutation that causes severe thrombosis. And that is an arginine 571 to serine mutation that is shown at the top of this, which may make space for this helix to move up and cause an increase in activation or activity of the factor IXA complex. And so lastly, what I'll say is that the other part of this interface that had never been observed before is how factor VIIIa recognizes exocyte 2 of factor IXA. And so within this factor VIII structure, we have these highly unstructured acidic regions. In this case, we call this the A2 peptide, which not only has acidic residues, but also has sulfated tyrosines. And so we see that this splays out across the exocyte 2 of factor IXA, which likely further supports stable interaction between the intrinsic tenase complex. So to conclude, I told you 3 separate mini stories of the work we've done in the last few years. The first of which is that we determined the structure of ET3 that not only showed the highest resolution structure of factor VIII to date, which can be a meaningful scaffold with which to do protein engineering, but it also allowed for us to re-refine the existing data sets for human factor VIII to improve those models as well. And we show that the ET3i, through our collaborators' evidence, is effective for this ex vivo lentiviral-driven gene therapy approach. I didn't talk a lot about all of the antibody structures that we've determined, except for one. But by and large, we see these commonalities that you might expect to exist for the epitopes, and that we often see buried hydrophobics, and almost always we see a lysine or an arginine that sits as one of the epitope hotspots. Okay? And then lastly, some of the data that I showed is for the intrinsic tenase complex and activated factor VIIIa, hopefully leading to more robust and reliable models of the intrinsic tenase complex that will be inspiring for the next generation of protein therapeutics for next generation treatments. So with that, I want to thank all of the students and postdoc. I had one postdoc work on this project over the last several years, so I want to thank all of them for their invaluable work. Our collaborators are shown on the right, and this work is largely funded by the NHLBI. Thank you for your time.
Clint, thank you very much for that. Did you mention that non-hemophiliacs, they develop spontaneous inhibitor development?
Non-hemophilia patients, yeah. So was that the question? Yeah. It's rare instances, sometimes happens in elderly people or postpartum women, where they will develop an immune response to factor VIII. We call that acquired hemophilia. That's often what porcine factor VIII has been clinically approved for, to my understanding. So either that or some other way. So, yeah.
That was beautiful. I'm curious about how factor 10 is complexing with factor VIII differently than factor 10 is complexing with its other cofactor as a substrate, tissue factor. Some of those lower resolution structures have been published. So what have you noted?
You're talking about prothrombinase structures?
No, I'm talking about 10As, tissue factor 10 with 7A versus what you're seeing with your intrinsic 10As.
I haven't done a robust modeling of factor 10 to this 10As complex yet, so it'd be hard for me to answer that question. But I will look into that.
Thank you.